Hello, welcome back for chapter 2 of My Stroke of Insight. My name is Dale, and I will be reading to you today, so I hope you enjoy this story time. For any two of us to communicate with one another, we must share a certain amount of common reality. As a result, our nervous system must be virtually identical in their ability to perceive information from the external world, process and integrate that information in our brains, and then have similar systems of output, including thought, word, or deed. The emergence of life was a most remarkable event. With the advent of the single celled organism, a new era of information processing was born at the molecular level. Through the manipulation of atoms and molecules into sequence, DNA and RNA sequences, information could be entered, coded, and stored for future use. Moments in time no longer came and went without a record, and by interweaving a continuum of sequential movements into a common thread, the life of the cell evolved as a bridge across time. Before long, cells figured out ways of hanging together and working together, which finally produced you and me. According to the American Heritage Dictionary, to evolve biologically means to develop by evolutionary process from a primitive to a more highly organized form. Earth's molecular brain of DNA is a powerful and successful genetic program, not only because it adapts to constant change, but also because it expects, appreciates, and takes advantage of opportunities to transform itself into something even more magnificent. It is perhaps of interest that our human genetic code is constructed by the exact same four nucleotides, which are complex molecules, as every other form of life on the planet. At the level of our DNA, we are related to the birds, reptiles, amphibians, and other mammals, and even to plant life. From a purely biological perspective, we human beings are our own species specific mutation of Earth's genetic possibility. As much as we would like to think that human life has attained biological perfection despite our sophisticated design, we do not represent a finished and or perfect genetic code. The human brain exists in an ongoing state of change. Even the brains of our ancestors of 2000 or 4000 years ago do not look identical to the brains of man today. Development of language, for example, has altered our brain's anatomical structure and cellular networks. Most of the different types of cells in our body die and are replaced every few weeks or months. However, neurons, the primary cell of the nervous system, do not multiply for the most part after we are born. That means that the majority of the neurons in your brain today are as old as you are. This longevity of the neurons partially accounts for why we feel pretty much the same on the inside at the age of 10 as we do at the age of 37 or 77. The cells in our human and the cells in our brain are the same, but over time their connections change based on their/our experience. The human nervous system is a wonderfully dynamic entity composed of an estimated 1 trillion cells. To give you some appreciation for how enormous 1 trillion is, consider this. There are approximately 6 billion people on the planet, and we would have to multiply all 6 billion people 166 times just to make up the number of cells combining to create a single nervous system, which is like still impossible to imagine. And that's dated back from whenever this book was. So there's more than 6 billion now. Of course, our body is much more than a nervous system. In fact, a typical adult human body is composed of approximately 50 trillion cells. That would be 8,333 times all of the 6 billion people on the planet. What's amazing is that this huge conglomeration of bone cells, muscle cells, connective tissue cells, sensory cells, etc., tend to get along and work together to generate perfect health. Biological evolution generally occurs from a state of lesser complexity to a state of greater complexity. Nature ensures her own efficiency by not reinventing the wheel with every new species she creates. Generally, once nature identifies a pattern in the genetic code that works towards the survival of the creature, like a blossom for nectar transmission, a heart to pump blood, a sweat gland to help regulate body temperature, or an eyeball for vision, she tends to build that feature into future permutations of that specific code, by adding a new level of programming on top of an already well established set of instructions. Each new species contains a strong foundation of time tested DNA sequences. This is one of the simple ways through which nature transmits the experience and wisdom bestowed by ancient life to her progeny. Another advantage to this type of built on top of what already works genetic engineering strategy is that very small manipulations of the genetic sequence can result in major evolutionary transformations. In our own genetic profile, believe it or not, scientific evidence indicates that we humans share 99.4% of our total DNA sequences with the chimpanzee. This does not mean, of course, that humans are direct descendants from our tree swinging friends, but it does emphasize that the genius of our molecular code is supported by eons of nature's greatest evolutionary effort. Our human code was not a random act, at least not in its entirety, but rather is better construed as nature's ever evolving quest for a body of genetic perfection. As members of the same human species, you and I share all but 0.01%, 1/100th of 1% of identical genetic sequences. So, biologically, as a species, you and I are virtually identical to one another at the level of our genes, 99.99%. Look around at the diversity within our human race. It's obvious that 0.01% accounts for a significant difference in how we look, think, and behave. Portion of our brains that separates us from all other animals is the outer undulated and convoluted cerebral cortex. Although other mammals do have a cerebral cortex, the human cortex has approximately twice the thickness and is believed to have twice the function. Our cerebral cortex is divided into two major hemispheres, which complement one another in function. Note, all of the pictures in this book have the front of the brain directed to the left. Whole human brain, cerebral cortex, right hemisphere, front of the brain, left hemisphere, back of the brain. Okay, so I'm going to show you the picture of this. The two hemispheres communicate with one another through the highway for information transfer, the corpus callosum. Although each hemisphere is unique in its specific types of information it processes, when the two hemispheres are connected to one another, they work together to generate a single seamless perception of the world. So take a look. It says, it's not focusing, so hopefully it will. All right, so if you see this is the back of the brain, that's the front of the brain, and you can see the two hemispheres, and they are attached in the center here by the corpus callosum. So I'll continue reading. When it comes to the intricate microscopic anatomy of how our cerebral cortices are finely wired, variation is the rule, not the exception. This variation contributes to our individual preferences and personalities. However, the gross or macroscopic anatomy of our brains is quite consistent, and your brain looks very similar to mine. The bumps, gyri, I'm guessing, somebody go ahead and let me know if I'm saying it wrong, and grooves, sulci, SULCI, of the cerebral cortex are specifically organized such that our brains are virtually identical in appearance, structure, and function. For example, each of our cerebral hemispheres contain a superior temporal gyrus, pre and post central gyri, a superior, superior parietal gyrus, along with a lateral occipital gyrus, just to mention a few. Each of these gyri are made up of a very specific group of they are made up of very specific groups of cells that have very specific connections and functions. For instance, the cells of the post-central gyrus enable us to be consciously aware of sensory stimulation, while the cells in the precentral gyrus control our ability to voluntarily move our body parts. The major pathways for information transfer between the various cortical groups of cells, called fiber tracts, within each of the two hemispheres are also consistent between us, and as a result, we are generally capable of thinking and feeling in comparative ways. All right, so I'm going to show you the picture of all of this stuff, and we'll see if it will zoom enough for you to see it. Okay, now I'm going to read through the labels of the brain. Post-central gyrus, sensory cortex. Precentral gyrus, motor cortex. Superior parietal gyrus, perception of physical boundaries. Lateral occipital gyrus, vision. Superior temporal gyrus, hearing and speech. Blood vessels supplying nutrients to our cerebral hemispheres also display a defined pattern. The anterior, middle, and posterior cerebral arteries supply blood to each of two hemispheres. Damage to any specific branch of one of these major arteries may result in somewhat predictable symptoms of severe impairment or complete elimination of our ability to perform specific cognitive functions. Of course, there are unique differences between damage to the right and left hemispheres. The following illustrations shows the territory of the middle cerebral artery of the left hemisphere, and this includes the location of my stroke. Damage to any of the middle cerebral arteries' primary branches would result in relatively predictable symptoms no matter who was having the problem. So take a look again at the picture. Okay, the middle cerebral artery, territory and major branches. So problems with movement, problems creating speech, problems understanding speech, problems with vision, and problems recognizing physical boundaries. The superficial layers of the cortex, which we see when we look at the external surface of the brain, are filled with neurons that we believe to be uniquely human. These most recently added on neurons create circuits that manufacture our ability to think linearly, as in complex language and the ability to think in abstract, symbolic systems like mathematics. Deeper layers of the cerebral cortex make up the cells of the limbic system. These are the cortical cells we share with other mammals. The limbic system, affect or emotion. Cingulate gyrus, ability to pay attention. Amygdala, fear and rage. Corpus callosum, okay, that's in the center, like we said. The hippocampus, learning and memory. And this is on the right hemisphere. It's showing it. Okay. I think they're just showing the right hemisphere, not that it's only located there. Okay. I could be wrong. The limbic system functions by placing an affect or emotion on information streaming in through our senses. Because we share these structures with other creatures, the limbic system cells are often referred to as the reptilian brain or the emotional brain. When we are newborns, these cells become wired together in response to sensory stimulation. It is interesting to note that although our limbic systems function throughout our lifetime, it does not mature. As a result, when our emotional buttons are pushed, we retain the ability to react to incoming stimulation as though we were a 2-year old, even when we are adults. Okay, that is very important to know about you and the people around you. As our higher cortical cells mature and become integrated in complex networks with other neurons, we gain the ability to take new pictures of the present moment. When we compare the new information of our thinking mind with the automatic reactivity of our limbic mind, we can reevaluate the current situation and purposely choose a more mature response. Okay? I'm going to read that section over again. As our higher cortical cells mature and become integrated in complex networks with other neurons, we gain the ability to take new pictures of the present moment. When we compare the new information of our thinking mind with the automatic reactivity of our limbic mind, we can reevaluate the current situation and purposely choose a more mature response. Okay, good news. That's great news. It might be of interest to note that all of today's brain based learning techniques used in elementary through high school capitalize on what neuroscientists understand about the functions of the limbic system. With these learning techniques, we try to transform our classrooms into environments that feel safe and familiar. The objective is to create an environment where the brain's fear slash rage response from the amygdala is not triggered. Primary job of the amygdala is to scan all incoming stimulation in this immediate moment and determine the level of safety. One of the jobs of the cingulate gyrus of the limbic system is to focus the brain's attention. When incoming stimulation is perceived as familiar, the amygdala is calm, and the adjacently positioned hippocampus is capable of learning and memorizing new information. However, as soon as the amygdala is triggered by unfamiliar or perhaps threatening stimulation, it raises the brain's level of anxiety and focuses the mind's attention on the immediate situation. Under these circumstances, our attention is shifted away from the hippocampus and focused towards self preserving behavior about the present moment. Sensory information streams in through our sensory systems and is immediately possessed or processed through our limbic system. By the time a message reaches our cerebral cortex for higher thinking, we have already placed a feeling upon how we view that stimulation. It is this pain or is this pain or is this pleasure? Although many of us may think of ourselves as thinking creatures that feel, biologically, we are feeling creatures that think. Okay, you feel first and then you think. Because the term feeling is broadly used, I'd like to clarify where different experiences occur in our brains. First, when we experience feelings of sadness, joy, anger, frustration, or excitement, these are emotions that are generated by the cells of our limbic system. Okay, so they're like immature, but still important. Second, to feel something in your hands refers to the tactile or kinesthetic experience of feeling through the action of palpation, touching. This type of feeling occurs via the sensory system of touch and involves the postcentral gyrus of the cerebral cortex. Finally, when someone contrasts what he or she feels intuitively about something, often expressed as a gut feeling, to what they think about it. This insightful awareness is a higher cognition that is grounded in the right hemisphere of the cerebral cortex. In chapter 3, we will discuss more thoroughly the different ways in which the right and left cerebral hemispheres operate. As information processing machines, our ability to process data about the external world begins at the level of sensory perception. Although most of us are rarely aware of it, our sensory receptors are designed to detect information at the energy level because everything around us, the air we breathe, even the materials we use to build with, are composed of spinning and vibrating atomic particles. You and I are literally swimming in a turbulent sea of electromagnetic fields. We are part of it. We are enveloped within it. And through our sensory apparatus, we experience what is. Each of our sensory systems is made up of a complex cascade of neurons that process the incoming neural code from the level of the receptor to specific areas within the brain. Each group of cells along the cascade alters or enhances the code and passes it on to the next set of cells in the system, which further defines and refines the message. By the time the code reaches the outermost portion of our brain, the higher levels of the cerebral cortex, we become conscious of the stimulation. However, if any of our cells along the pathway fail in their ability to function normally, then the final perception is skewed away from normal reality. Our visual field, the entire view of what we can see when we look out into the world, is divided into billions of tiny spots or pixels. Each pixel is filled with atoms and molecules that are in vibration. The retinal cells in the back of our eyes detect the movement of these atomic particles. Atoms vibrating at different frequencies emit different wavelengths of energy. And this information is eventually coded as different colors by the visual cortex in the occipital region of our brain. The visual image is built by our brain's ability to package groups of pixels together in the form of edges. Different edges with different orientations, vertical, horizontal, and oblique, combine to form complex images. Different groups of cells in our brain add depth, color, and motion to what we see. Dyslexia, whereby some written letters are perceived in reverse from normal, is a great example of a functional abnormality that can occur when the normal cascade of sensory input is altered. Cortical organization, and here's another image that I will show you. Frontal region, it involves self motivation, appropriateness of behavior. Parietal region is the integration of all sensory information. Occipital region is vision. Temporal region is hearing, learning, and memory. Similar to vision, our ability to hear sound also depends upon our detection of energy traveling at different wavelengths. Sound is the product of atomic particles in space colliding with one another and emitting patterns of energy. Okay. So it seems like everything is wavelengths, frequencies, and patterns of energy. Okay. So we'll, see the importance of that as we continue to learn through many different books. All right. The energy wavelengths created by the bombarding particles beat upon the tympanic membrane in our ear. Different wavelengths of sound vibrate our eardrum with unique properties, similar to our retinal cells. The hair cells of our auditory organ of corti translate this energy vibration in our ear into a neural code. This eventually reaches the auditory cortex in the temporal region of our brains, and we hear sound. So let me just pause real quick to explain this. That what we see in our visual surroundings, it goes to our retinal cells, which is like the wallpaper on the inside of your eyeball, and then it translates it into like an energy frequency that transmits it into your brain for understanding. Okay, it's not like your eyes are seeing directly into your brain and your brain is understanding. We're not hearing sounds per se. It's an energy collision and patterns and frequencies that are vibrating the tympanic membrane in your ear, and then it's transferring it into your brain as like a it's like an energetic transmission, okay? So it's not as simple as everything seems. It's not like what we see is what we see and what we hear is what we hear. It seems so simple, but it's not that simple. Okay, that's why when you learn about these things, you can learn to understand why energy and frequency are important in creating changes in your world, because your brain, it sees what's in your mind's eye just as well as it sees what's in your visual perspective. Right. Our most obvious abilities to sense atomic/molecular information occur through our chemical senses of smell and taste. Although these receptors are sensitive to individual electromagnetic particles as they waft past our nose or titillate our taste buds, we are all unique in how much stimulation is required before we can smell or taste something. Each of these sensory systems is also made up of a complex cascade of cells, and damage to any portion of the system may result in abnormal ability to perceive. So that could be like from the anatomical structure or in the brain. Okay. Finally, our skin is our largest sensory organ, and it is stippled with very specific sensory receptors designed to experience pressure, vibration, light touch, pain, or temperature. These receptors are precise in the type of stimulation they perceive, such that only cold stimulation can be perceived by cold sensory receptors, and only vibration can be detected by vibration receptors. Because of this specificity, our skin is a finely mapped surface of sensory reception. The innate differences we each experience in terms of how sensitive we are to different types of stimulation contribute greatly to how we perceive the world. If we have problems hearing when people speak, then we will only hear bits and pieces of conversation and make decisions and judgments based upon minimal information. If our eyesight is poor, then we will focus on fewer details and our interaction with the world will be affected. If our sense of smell is deficient, then we may not be able to discriminate between a safe environment and a health hazard, rendering us more vulnerable at the opposite extreme. If we are oversensitive to stimulation, we may avoid interacting with our environment and miss out on life's simple pleasures. Pathology and disease of the mammalian nervous system generally involves the brain tissue that distinguishes that specific species from other species. Consequently, in the case of the human system, the outer layers of our cerebral cortex are often vulnerable to disease. Stroke is the number one disabler in our society and the number three killer, because neurological disease often involves the higher cognition layers of our cerebral cortex. And because stroke occurs four times more frequently in the left cerebral hemispheres, our ability to create or understand language is often compromised. The term stroke refers to a problem with the blood vessels carrying oxygen to the cells of the brain. And there are basically two types: ischemic and hemorrhagic. Okay. According to the American Stroke Association, the ischemic stroke accounts for approximately 83% of all strokes. Arteries carry blood into the brain, and their shape tapers smaller and smaller as they travel farther away from the heart. These arteries carry life supporting oxygen necessary for the cells, including neurons, to survive. With ischemic stroke, a blood clot travels into the artery until the tapered diameter of the artery becomes too small for the clot to pass any further. The blood clot blocks the flow of oxygen rich blood to the cells beyond the point of obstruction. Consequently, brain cells become traumatized and often die. Since neurons generally do not regenerate, the dead neurons are not replaced. The function of the deceased cells will be lost permanently unless their neurons adapt over time to carry out their function. Because every brain is unique in its neurological wiring, every brain is unique in its ability to recover from trauma. Ischemic blood clot, an artery is blocked and oxygen cannot get to the cells. So here is what it looks like. So when she's talking about it being tapered, right? It's just like going from wider to narrower. That is the tapered effect. So all these blood cells can flow through just fine, or blood clots or whatever they are. But if one gets stuck, then the blood cannot move beyond it. Okay. So oxygen cannot move beyond it. A hemorrhagic stroke occurs when blood escapes from the arteries and floods into the brain. 17% of all strokes are hemorrhagic. Blood is toxic to neurons when it comes in direct contact with them. So, any leak or vascular blowout can have devastating effects on the brain. That is kind of crazy to think that blood is toxic to neurons, like to the brain when it's coming from outside of the blood vessels. Okay. One form of stroke, the aneurysm, forms when there is a weakening in the wall of a blood vessel that consequently balloons out. The weakened area fills with blood and can readily rupture, spewing large volumes of blood into the skull. Any type of hemorrhage is often life threatening. So, a hemorrhage is bleeding. If you don't, so like the hemorrhagic stroke is when there's bleeding that like busts through the blood vessels and out into the brain, the neurons. Weakened area fills with blood, and okay, we already read that. Fills with blood and can readily rupture, spilling large volumes of blood into the skull. Any type of hemorrhage is often life threatening. So, an aneurysm, thin wall of blood vessel ballooning out. Oh no, it's going to blow, is what it says. Normal thick wall of the blood vessel is okay. So, in this picture, you can see like the walls of the blood vessel are supposed to be thick. And this is when there's like a weakness in that wall. And so the blood moves into this portion where it does not belong. It's ballooning out because of the pressure going through that area. An intravenous malformation, AVM, is a rare form of hemorrhagic stroke. It is a congenital disorder whereby an individual is born with an abnormal arterial configuration. Normally, the heart pumps blood through arteries with high pressure, while blood is retrieved through the veins, which are low pressure. Okay, so arteries, high pressure, veins, low pressure. A capillary bed acts as a buffering system or neutral zone between the high pressure arteries and the low pressure veins. Normal blood flow, artery, capillaries, veins. So you can see it in this picture. In the case of an AVM, an arteriovenous malformation, an artery is directly connected to a vein with no buffering capillary beds in between. So the capillaries are the thin little areas right there, those, the blood vessels that are thinner. Um, Over time, the vein can no longer handle the high pressure from the artery, and the connection between the artery and vein is broken, spilling blood into the brain. Although the AVM accounts for only 2% of all hemorrhagic strokes, it is the most common form of stroke that strikes people during their prime years of life, ages 25 to 45. I was 37 when my AVM blew. Okay. Okay, so here is a picture of an arteriovenous malformation, AVM. So you can see where there should be capillaries in between there. It's directly linked from the artery to the vein, and the artery has high pressure and the vein has low pressure. So you may be able to withstand that for a long time, and then it just, again, only 2% of the population. So, or 2% of strokes are this type. So don't get nervous about everything you hear about, all right? Just understand what causes it. Regardless of the mechanical nature of the vascular problem, be it a blood clot or a hemorrhage. No two strokes are identical in their symptoms because no two brains are absolutely identical in their structure, connections, or ability to recover. At the same time, it is impossible to talk about the symptoms resulting from stroke without having a conversation about the innate differences between the right and left cerebral hemispheres. Although the anatomical structure of the two hemispheres is relatively symmetrical, they are quite diverse in not only how they process information, but also in the types of information they process. The better we understand the functional organization of the two cerebral hemispheres, the easier it is to predict what deficits might occur when specific areas are damaged. Perhaps more importantly, we might gain some insight into what we can do to help stroke survivors recover lost function. Okay, here are warning signs of a stroke, and it's a nice little I can't think of what it's called. A little memory trigger where it's like a word and then each letter stands for something. Acronym. Yay. Okay. So S stands for speech or any problems with language. T stands for tingling or any numbness in the body. R is remember or any problems with memory. O, off balance, problems with coordination. K, killer headache. E, eyes or any problems with vision. Stroke is a medical emergency, call 911. Okay, so that is the acronym. Let us see. Yeah, that is the end of chapter 2. Okay, come on back for chapter 3.